Biodiversity is one of the most intricate and beautiful features of our planet. Life takes on a variety of astonishing shapes, forms, and strategies for survival. Mollusks, in particular, the second most diverse phylum of animals on the planet, uniquely represent this amazing variety. From the largest living invertebrate, the colossal squid, to snails which have colonized nearly every habitat on the planet, to tiny worm-like animals that can sometimes be as small as a grain of sand. They epitomize this amazing variability. Biodiversity is often discussed in terms of ecosystem services like food production, climate regulation, or medicine. And while these things are important, they aren't why I study biodiversity. I study it because I believe that biodiversity has intrinsic value. Species do not need to justify their existence by being useful to humans. They represent millions of years of evolutionary history and have a right to exist independent of us. But before we can protect biodiversity or even appreciate it, we first need to know what biodiversity exists. My research focuses on a little-known class of mollusks called Solenogasters. They're worm-like mollusks that live on the ocean floor, and instead of having a shell like many other mollusks, they're covered in scales or spines called sclerites. Globally, only about 330 species of Solenogasters have been formally described. That number alone suggests rarity and low diversity. But my research tells a very different story. I'm studying more than 2,000 specimens collected across the northern Atlantic over the past 2 decades. This collection is one of the most comprehensive regional samplings ever assembled, providing an unprecedented opportunity to understand Solenogaster diversity. Over the last several years, I've been sorting this collection into morphospecies. One of the most important characters for doing this are the sclerites. I can scrape these sclerites off of the body using a small needle and view them under a compound light microscope. I can also take a piece of the mid-body and image it using the scanning electron microscope. Here we can see a comparison when we view it under a normal light microscope, and now the scanning electron microscope. You can really see the sclerites using this. This collection has yielded some incredible sclerite diversity. I can then extract DNA from that same mid-body piece that I used for scanning electron microscopy and test our morphospecies hypothesis using DNA barcoding. We do this by sequencing a couple of genes. These sequences are composed of A's, T's, C's, and G's, representing the 4 nucleotide bases that make up DNA. We can align the sequences between each specimen or species and compare them to see how similar they are to one another, and use these to build phylogenetic trees, which model the relationships between these animals. Through this work, I've identified over 165 distinct species. I estimate that 85% of these likely represent species that are new to science. In other words, in just one region of the world, we may be increasing the known diversity of this class of animals by almost 50%. I've already described 9 of these new species, but there's an issue. Describing a species, particularly in this group of mollusks, is a very time-intensive process. The first steps include embedding the head and tail end in paraffin wax, which is kind of like a candle wax. Then, we section the animal using a microtome. This is basically a glorified meat slicer like you'd find at your local deli, but the slices are much thinner. We then stain these sections with dye that make the different tissues easier to see. Then we look at each section, one after another, and meticulously reconstruct their internal anatomy. The shape and arrangement of these organs can be important for differentiating species. I think it's imperative that people engage with the biodiversity around them. While my Solenogasters may be difficult to engage with for most, as they're tiny and found in the deep sea, mollusks generally are a great entry point for citizen scientists to engage with the world around them. One of the main reasons for that is that most have shells. Almost anyone that's gone to the beach has picked up a shell and admired its beauty, or wondered about the creature that created it. For this reason, I founded a nonprofit called The Naturalist Project. Our website showcases free-to-use seashell identification guides for the states of the eastern US and Gulf of Mexico, figuring over 550 species. We also produce videos on our YouTube channel showcasing the amazing biodiversity of our planet, from things that you can find in your backyard to strange wonders from the depths of Antarctica. I believe that the more people that engage with the biodiversity around them, the more they will connect and begin to care about it. No matter where it's found and no matter how small the animal may be, the biodiversity of our planet is intricate and beautiful. It's important that we don't forget to slow down our busy lives occasionally, to observe and explore the world around us, and appreciate the stunning array of life that can be found on this planet.